Hello, everybody, and welcome to part one of chapter 28. So we're just now starting chapter 28 with this lecture, and we're probably going to have 3 or 4 different parts to this lecture on chapter 28, I suppose. But this time, for this chapter, we're going to cover all of the protists, and there's certainly a wide variety of different protists out there. So there's a lot to cover in this chapter. A lot of really interesting things as well that we'll be looking at. And so protists then are very small generally. They're larger than prokaryotes, but still almost all protists are microscopic as well. But there's a great variety of different organisms in a drop of pond water or slough water, as we would say locally. And a lot of those living organisms in that slough water would be protists. So protist, kind of an informal name, and it's a group of mostly unicellular eukaryotes. So they've all got the eukaryotic cells then, so cells that do have a nucleus and a number of organelles, and their chromosomes come in pairs as well, and a lot of eukaryotic systematics. Systematics is basically a way of classifying organisms, and also in a way of it's a way of looking at what the different putting organisms into groups and kingdoms, phylums, classes, orders, etc., and figuring out what the relationships are to different groups, and whether groups are closely related or distantly related, trying to figure out what those relationships are. And so recent work in systematics has actually changed the classification of these things a bit long, and protista is no longer a kingdom. But of course, it's no longer a kingdom. What is it? It's still a group, kind of an informal group, basically. And you can see. So here is basically a typical protist, and these are actually prokaryotes, and you can see that in general protists are larger than prokaryotes. This one has got some crazy grooves on the surface of it, and these photographs are taken by a scanning electron microscope, and this one is kind of shaped like an acorn, a butternut squash, actually. Kind of interesting. So there's a lot of wide variety of different sizes and shapes, and here's trumpet-shaped protist called stentor, and so a lot of different types of sizes and shapes, and also amongst this group, amongst protists, there's a wide variety of different types of feeding strategies as well, and different habitats that they're in and stuff. So they have organelles, and they're generally more complex than prokaryotic cells. And so the organisms in most eukaryotic lineages are protists, and most protists are unicellular. And they have more structural and functional diversity than any other group of eukaryotes. They don't have more species than like the arthropods, for example, but they have more structural and functional diversity, though. And we'll look at all of that, the whole gamut of that diversity in this chapter. And though most of them are unicellular, there are some that are colonial in colonies, just like we saw with protists, and there are some that are multicellular and can get quite large, like the seaweeds and kelps. and we'll see those later on in the chapter. But they can be very complex, and because all biological functions, just think of this, all biological functions have to be carried out by that one cell, by the organelles in just one cell. They are the most nutritionally diverse eukaryote groups. So there are a lot of photoautotrophs that have chloroplasts that have the photosynthetic pigments in them, but there are heterotrophs as well that absorb organic molecules or maybe ingest larger food particles, and maybe whole cells. Then there are mixotrophs, interesting mixotrophs. They combine photosynthesis and heterotrophic nutrition. So a good mixture then. And some will reproduce asexually. So they just divide the cell into two cells, while others will reproduce sexually and exchange genetic material between two different individuals. Or they'll reproduce by the sexual processes of meiosis and fertilization. Our understanding of these relationships is changing all the time. Several hypotheses have been proposed and discarded, and there's a lot of academic research and investigation of this group. One current hypothesis divides all eukaryotes into four supergroups. Four supergroups then. So the excavata, the archaeoplastida, the SAR, and the uniconta. And so let's, and then here's an extra picture just thrown in for a good measure. And so we'll look at these one at a time. Excavata, so look at these weird shapes and colors and designs of all of these different groups. And there's a lot of videos here, so you can see a video of a Volvox colony kind of moving around. And here's another Volvox daughter colony being produced. And all sorts of videos. So Volvox are colonial. They're individual cells, but they're stuck together in a colony, and they work somewhat. Here's an inversion. Sorry, Volvox colony inversion. Here's another inversion, and producing sperm. Volvox sperm and the female cell. Oh, look at that! It's almost got crushed by that sphere that moved, that spherical colony that moved by. So you can watch those videos and enjoy them on your own time. And meanwhile, we will push on then through the lecture and through the chapter. So evolutionary hypothesis that much of the protist diversity then was actually caused by endosymbiosis. And I think we talked about endosymbiosis in the last chapter, but endo means inside, and symbiosis, remember, means two different species, members of two different species living in a close relationship with each other. But endosymbiosis is where one organism of one species lives inside the cell or the cells of another organism from another species. And so the host is the larger cell. And they think that mitochondria, you remember, mitochondria and plastids, very large, complex organelles. You probably remember those from General Biology 1. But they think that maybe these mitochondria and plastids were originally endosymbionts. So endosymbionts are just the little organisms that are living inside the symbiotic host. So they may have originally been endosymbionts. And you can see here's some sort of heterotrophic eukaryote, and then a cyanobacterium, so a bacteria, a prokaryote. A cyanobacterium actually enters or is taken in by this whatever type of heterotrophic eukaryote. And so there's endosymbiosis going on. The cyanobacterium is living inside as an endosymbiont, and it's got 3 different membranes to it. And one of the membranes becomes lost in the red and green algae descendants. And so, maybe evolutionarily, one branch of the family develops into red algae, and others develop another branch that develops into green algae. And maybe this red alga becomes an endosymbiont inside another cell. So you've got an endosymbiont. So now you've got an endosymbiont within a host, but that host is an endosymbiont within a larger host. And that might develop into, maybe some of these endosymbionts develop into plastids in one branch, and maybe another branch develops differently, and maybe those endosymbionts are mitochondria or something. And here's, more endosymbiosis takes place, more secondary endosymbiosis takes place maybe, and developing into other organelles then. So, this is a proposed hypothesis then for development that might have taken place. And so let's look at these 4 different groups. First of all, the clade, the group called excavata. And it's called excavata because of its kind of unique cytoskeleton. Some members of this group have an excavated, so it's called excavata because they have an excavated feeding groove on one side of the body. And so these excavates, members of the excavata, the excavates then have 3 basic monophyletic groups. Then the diplomonads, the parabacillids, and the euglenozoans. So eugleno because they look like the euglenids, and zoa actually means animal in general. Zoa means animal in general. And so here's the group excavata, and you got the diplomonads and the parabacillids that branched off from each other, and the euglenozoans that also branched off as well at some point, they think. So basically, the diplomonads and the parabacillids, they lack plastids, don't have any plastids, and they have kind of a modified mitochondria, and most are actually in anaerobic environments. So, do you remember what is an anaerobic environment? Do you remember what an anaerobic environment is? You've got aerobic environments and you've got anaerobic environments. So anaerobic environments are environments without oxygen. So the diplomonads then, what's unique about them? They've got a reduced mitochondria. They're called mitosomes. They get energy from anaerobic biochemical pathways, like maybe fermentation, remember, from the last chapter. They have 2, now this is really stranger, 2 equal-sized nuclei. And so, diplo means 2 generally. So 2 equal-sized nuclei and a lot of flagella, multiple flagella on their body. They're often parasites, like Giardia intestinalis. It causes giardiasis, intestinal disease. Giardiasis is an intestinal disease that you usually get from drinking water that's polluted, maybe, and it has Giardia intestinalis in it. Or there's also Giardia lamblia that will cause human disease. And then the parabacillids also reduce mitochondria that are called hydrogenosomes, that generate some energy anaerobically without oxygen. And so Trichomonas vaginalis then, trichomonas disease, sexually transmitted parasite that causes trichomonas. And here's Trichomonas vaginalis, and it's this is photographed through a scanning electron microscope. They've got undulating membrane along the outside, and we'll see others like that as well. So flagella and also undulating membranes that help them move. And then the euglenozoa, kind of diverse, a large variety. There are predatory heterotrophs. There are photosynthetic autotrophs. There are also mixotrophs and parasites of various types. So the main feature then distinguishing them is a spiral or crystalline rod. So you got to look at the flagella in detail, and you'll see a spiral or crystalline rod in the flagella, and we'll see a picture in just a minute. It includes kinetoplastids and the euglenids. And so here's, if you look at so this one basically, this euglenozoan has 2 flagella, and if you look at the flagella in detail in cross section, there's actually crystalline rods, cross section of a crystalline rod, and some microtubules as well, but crystalline rods then in those flagella to give a stiffness and support to the flagella. So the kinetoplastids, then they have a single mitochondrion inside the cell with an organized mass of DNA inside that mitochondrion called a kinetoplast. They're basically free living, not endosymbionts and not parasitic. Basically free living then, the free living ones that are free living then consume prokaryotes in freshwater, marine, and moist terrestrial areas. There are some that are parasites, though. Either they parasitize animals, plants, or other protists. Good example then, kinetoplastids in the genus Trypanosoma or Trypanosoma, some people call them, that cause sleeping sickness and other things in humans. Another pathogenic trypanosome causes what's called Chagas disease, which used to be in just Central and South America, but now there's actually a lot of Chagas disease in the United States as well. And it's produced by the bite of the kissing bug, believe it or not. You heard it here first. Chagas disease caused by the kissing bug. And so see here, here's undulating membranes then on these trypanosomes. So the trypanosomes are in kind of a light blue, and then the human red blood cells, of course, are in kind of a reddish color. So they evade or avoid the host immune system by producing proteins on their surface that are different, with a different molecular structure in each generation. So the cell surface proteins change each generation, and so they can. So basically, even if the host immune response keys in on their particular cell surface proteins, well, in the next generation, their cell surface proteins are going to have changed, and so the immune system won't be able to recognize them in the next generation. The euglenids, then, they have either 1 or 2 flagella that come out of like a little pocket at one end of the cell. Some of them are mixotrophs, so they can they can live as either autotrophs or heterotrophs depending on environmental conditions. So it's kind of nice, and they're kind of shapeshifters. They can either be autotrophic or heterotrophic depending on what conditions they're living in. And so here's a good example of a euglena. Then there's one long flagellum. There's actually an eye spot. So here's the actual photograph of it, and then here's the diagram of it. So there's an actual eye spot, so they can actually, they can't detect an optic image from it, but they can tell which direction the light is coming from. And so they've got chloroplasts and nucleus and contractile vacuoles. Whoops. Excuse me there. Contractile vacuoles that you may remember from, you probably covered those then in General Biology 1. And there's some cool videos showing how they move. And this next video shows you not only how it can move, but how flexible the body is. Look at that. It's as flexible as a leech almost. But this is just a single cell. So kind of a cool type of protist. So now the next group is called SAR. SAR, and it's named for the first few letters of the major groups, the major clades in this group then. So S is for straminophiles, the A is for alveolates, and the R is for rhizarians. One of the most controversial groups, not controversial in what they do, but controversial according to how to classify them. So we've got straminophiles, they're diatoms, gold algae, and brown algae. The alveolates, they're dinoflagellates, apicomplexans, and ciliates. And malaria is a ciliate. And they're rhizarians as well. They're radiolarians, the forams, and the cercozoans. Here's that term zoa again, meaning animal-like. So the SAR group then. First of all, then the straminophiles. Straminophiles then, it's some of the most important photosynthetic organisms that we have on Earth. Most have a hairy flagellum paired with a smooth flagellum. And so diatoms are in this group, and also the golden algae, so-called golden algae, and the brown algae as well. So here's the smooth flagellum, and here's the hairy flagellum. Kind of interesting things then. Diatoms basically are unicellular algae. They have a two-part glass-like cell wall made of silicon dioxide then, kind of a tough, unyielding two-part glass wall. And so it's kind of like two parts of a petri dish that fit together. And they can be a major component then of the phytoplankton. Remember now, plankton are small, usually microscopic things in the water, and phytoplankton are the ones that are photosynthetic, like plants. Highly diverse, so a large variety of different types. This is really cool then, a cool diatom photographed by scanning electron microscope. So this is Triceradium morlandii. And then a few other, and you can see, so it's really nice videos showing how they move and things. Now this is an oscillatoria, I think, but these brownish yellow ones are the diatoms. And here's some more movement of diatoms. See, here's long and thin diatoms and kind of football-shaped diatoms. And here's another one over here, moving over here. Behind, I think this might be oscillatoria, I'm not quite sure. You might remember oscillatoria from the last chapter. So After a diatom bloom, that is, after a huge increase in diatom populations, many of the dead ones follow the ocean floor where the decomposition is slow, and the carbon that they took up from the atmosphere and incorporated into their biomass is sequestered, that is, kept in a like a more or less permanent solid form, sequestered on the ocean floor for decades to centuries. Then, and so sometimes scientists think, well, maybe this is a way to remove excess carbon dioxide. Maybe we should fertilize the ocean with iron and promote large diatom blooms, large increases in diatom population, and that will facilitate then movement of carbon dioxide to the bottom of the ocean and sequester it there for a good long time, at least till we can figure out what to do with it. And then the golden algae, remember the golden algae are part of this group as well. They're named for their color then. It's a mixture of yellow and brown carotenoids. Carotenoids are colored pigments, and orange carotenoids are what give carrots their orange color. And carotenoids can be used in photosynthesis as well in some species. They're typically biflagellated, have two flagella, and they're all photosynthetic, but some are mixotrophs. Most are unicellular, but some are actually colonial and can get to a fairly good size. Here's a colonial golden algae then. It looks like a bouquet of flowers. And it's got a flagellum and inside an outer container. And very colorful, very nice. And this is a regular microscope, regular light microscope for this photograph. Then the brown algae, the largest and most complex of the algae then. All of them are multicellular, and most are in salt water. And they include what we commonly think of as seaweeds or kelp, or kelp is in this group as well. KELP, the kelp. And we'll see pictures in just a minute here. Oops. So they have plant-like structures. Then they have a root called a holdfast that anchors it to whatever substrate it's in, and then a stem like stipe, they call it, that supports different things that are kind of like leaves. They're called blades, and some of them have gas-filled bubble-shaped floats to keep the photosynthetic structures near the water surface, so that they can. So if you've got a float and that's keeping your photosynthetic structures near the surface, that's a good thing, because near the surface you get most of the light. But unlike plants, they lack true tissues and organs, so they're not actually a plant because they lack true tissues and organs. And here's a good photo of them. But some of these can be 20 feet long, though, really sizable. And there's alternation of generations as well in this group, but I don't think I'll get into that now. I think I will cover that during our next lecture, our next segment of these series of lectures then on chapter 28 on the protists. So, but remember though, always check your email and your Canvas each day, at least once a day, for important announcements and things, so to keep up with what's going on in the class. So anyway, I'll see you later then in part 2 of chapter 28, talking about the fun and diverse protists that are out there.